My name is Garrett Ride.
And I'm Harrison Oakes.
We're both undergrad students at Brigham Young University, and we did our poster on enabling better interoperability and reusability for Down syndrome transcriptomic datasets. In recent years, with advancement in technology, transcriptomic datasets or gene expression datasets have become more accessible. As we gather more datasets on gene expression, we're able to better understand genetic diseases, their origin, and possible solutions for treating these in the future. However, researchers can either create new datasets in new experiments, which takes time and resources, or they can reuse past experiments and past datasets. However, this poses a problem as these datasets are coming from many different sources. It's hard to reuse the datasets and hard to combine these datasets. And when different researchers might not have the technical skills to be able to solve this problem or the time to solve this problem, we decided to create this pipeline to do it for them. So as we gather datasets from all over the country and they go through this data pipeline, they'll come out in a way that will be reusable and will be interoperable, meaning we'll be able to combine datasets.
Which brings us to our figure 1. Our figure 1 is an example of our entire pipeline from the start, which is collecting the data, to the end, which is outputting data that can then be combined. So to start, we have a little example of taking different data sets from different researchers, whether that's from a university or any type of research. We take it and we then feed it into our pipeline. From there, it will then do 3 different checks. First, quality control, second, normalization, and third, metadata standardization. And then from there, it will basically standardize each data set, so then researchers can come back and combine these data sets or combine specific ones, whatever they choose. And our expected product is going to be what this little example here shows, something that somewhere researchers can go to and pick and choose the data sets that they want, or they can choose the entire mixture of data sets and be able to do research on them, improving results and finding better ways to improve the lives of individuals with Down syndrome.
Yeah, so the first step is data collection. And as researchers from different places are collecting their data, they often make their data available on a gene expression omnibus. We are using R for our research, and R has a specific package called GeoQuery. GeoQuery can access these data sets that are on gene expression omnibus and can download these data sets. So after we download the data sets, we then move on to quality control. Many of the samples within a data set will be considered low quality because maybe someone had a finger on a sample, which will then make it not helpful for researchers in the future. So in order to test if a specific sample is low quality, we had 3 methods of finding outliers. If one sample is found to be an outlier in all 3 methods, then we marked it as poor quality and remove it from the data set. In this figure, figure 2, we're showing for each data set the percentage of the data set that is low quality. And as you can see, most of them are at 0%, meaning most of them, all of their samples are good quality. However, there's a few goes all the way up to about 15% of one data set that we had to remove.
Which then brings us to normalization and mapping. Normalization, we're using a package called scan.upc, which will basically, our goal is to get rid of any biases. For example, a guanine cytosine bias, to keep all the data similar and normalized. And then from there, mapping, our goal is to filter out the low quality probes and summarize the data at the gene level. And then finally, we will standardize the metadata. So with all of these different research groups that are doing the research on this, a lot of the headers are labeled differently. So for example, one is for the gender of the species. Some researchers are using F, some are using FEM with a period, some are using capital F, some are using female spelled out, and the computer doesn't know what to do with this. And so what we're doing is we're using a standardized ontology to standardize all of the metadata used in all the different data sets, which will then output our expected product, which is a standardized data set, including the metadata and the data itself. So then other researchers, including ourselves, can come in later and pick and choose which data sets they want to use, or they can choose the entire data set, which will then give better numbers and better quality data. Thank you.